So, thanks very much, everybody. Open to questions. Rachel.
So I have a question that might be relevant for Waropon and Ewan. With the unbiased transcriptomic approaches, sometimes the results can be somewhat overwhelming in terms of the number of pathways that you get back. Where do you start, and how do you go about, is it a CRISPR screen? What strategies do you both have in mind for taking some of these pathways forward?
What I start, right? Actually, firstly, we focus on upregulated gene first at different stages because we, personally, I think that this is gonna, is important gene that could link to the important pathway with the high expression of the gene. Yep.
So I think in the case of my data set, I'm in a somewhat unique position where because we sequenced both wild types and the mutants prior to stretch and post-stretch. So we can do differential gene expression analysis in response to stretch in both, and then try and find unique population or unique genes to the mutants. It still means we have a lot. But yeah, that at least allows us to narrow it down somewhat.
Yeah, and then it's identifying which of those are just passenger things that happen anyway, and then which are actually driving the biology of it. So anyway, wish you all the best for taking your targets.
Could I just continue that? That's our point there, actually. So you're stretching, you're mimicking the stretching of cells and seeing what happens. But is this chicken or egg? What is, why do cells get stretched anyway? Is it the primary pathology? Is the stretching in itself a passenger?
Yeah, it's definitely a chicken or the egg, or certainly can be. There is a world in which we've got P53, P10 in these organoids, or in vivo you've got a proliferative advantage that is being conveyed through mutation, and does that in itself, drive this neoplasia and then creates the mechanics. Will we answer that? Maybe. I've got plenty of time. I've still got another 2 and a bit years.
It goes quickly.
Yes. So I don't know, but I think the fact is, we can create what I think are interesting biomechanical signals within our mutant group, but we also see interesting biomechanical signals within the wild type group. So there is still mutation on mutation. If you're in that environment, it still might generate signals that are important in initiation.
Excellent. And sorry, it's a little tiny bit more. You have your transcriptome to measure the output. Are there any structural ways in which you're looking at the stretching as well? There are some antibodies, aren't there?
So in terms of, sorry, can you repeat the question?
Just how are you measuring the stretching?
Yeah, so there is, we've been looking into using a fluorescence lifetime reporter called Flipper, which will give us readouts on membrane tension, which is interesting. There are some FRET systems for nuclear envelope tension. There are definitely avenues for us to take, and I think it's something as part of establishing this model, for me, is something that's really important to understand. Really
nice esophageal data using the monoclonals. Very nice. Okay, brilliant. Sorry.
So a fairly soft question, I think, for you, Sheila. You showed some really nice combination of data in cell lines and then in the xenograft models. Are you interested at all in looking into, or do you think it'd be a good idea to look into genetically engineered mouse models and see how they respond to your drug combination? And maybe the reason it came to mind is because you showed or suggested a number of pathways downregulated, and whilst it's not a perfect model by any means, I'm also wondering if you could utilize hydrodynamics to test various genetic combinations and show that actually it is a drug combination that's effective on multiple pathways and in multiple genetic contexts, if that makes sense. So I just wondered if you're interested in looking at more complex mouse models, or and where the combination's going, I guess.
I think I think I'm quite interested in looking at a syngeneic model because I think niclosamide has other effects on the immune system as well. So I'd be quite interested to be able to look at that. But I don't want to continuously do different mouse models because the purpose of this was to be able to translate something a little bit faster. So, I think, yes, there is a place for looking at models and mechanisms.
That's my basic scientist self showing through. I just want to interrogate it further. I think syngeneic models are a great idea as well,
Yeah.
Ewan, can I ask a question? This might sound super simplistic. Yet, as a consequence of stretch, majority of genes downregulated, some upregulated. Those upregulated include the mismatch repair genes.
Yes.
Is stretch good or bad?
Yeah, it's a good question. I think I'm going to give a very hand-wavy answer in terms of I think it's too early for us to know, but as we can see, so when we stretch the organoids, the wild types don't stretch, first of all, to the same size that the mutants do. So I think there's an underlying difference that mutation allows you to respond to these things differently. So that is something interesting. There could be a world in which either downregulation or the mismatch repair is that stretch in itself induces DNA damage that if cells can survive it, then it drives that process of mutational acquisition. This is with the huge caveat that this is in our stretch, and this is happening on a much shorter timescale than we'd expect in vivo. So I guess one of the questions is the DNA mismatch repair something that we would still see in a more in vivo setting? I don't know.
So stretch is bad.
Oh no.
Okay,
Catch
put it
me
another
after.
way. Stretch is part of the sort of oncogenic process.
Yeah, I mean, so I think the downregulation DNA damage is something that we're interested in having a look at. So doing H2A staining and seeing what's actually happening, because the KEGG term says that there's DNA damage, but it doesn't mean it's actually occurring. So there is that caveat, but as well, there is a lot of, if you're a cancer cell, there is a lot of stuff, especially within the RPPA, that is incredibly beneficial for you as a cancer cell. I think, as all things are, it's a matter of balance.
Does the model, if you stop treating with the drug that induces the swelling, does it reverse, and then can you show reversion or a third state?
So we've taken the stretch model out to 24 hours, and you sort of reach a peak of stretch around 16, but they will keep stretching to 24. We have not tried what happens when we take it off, but the paper that it was based on was in intestinal organoids, and after, I can't remember the time frame, but they start to revert back. I don't know if they go back to the original size, but yeah.
Sheila?
Can I ask a question? So how does stretch relate to stiffening? As in, when you've got stiff breast tissue, you get the stiffening, don't you?
Mm-hmm.
How does that relate? And is it to do with collagen deposition or anything like that?
Yeah, so there's definitely an interesting space in which we could look at. So we're stretching these organoids within matrigel, which has collagen, fibronectin within it. There is a world in which we could change the stiffness of the environment around these organoids. The primary way people tend to do that is with hydrogels, and cross-linking with ribose to generate stiffer and stiffer sort of ECMs within there. Not something we've done yet, but again is something that's interesting because we do know that these environments are stiffer in neoplasia anyway. I hope that answers your question.
Yeah, and then you saw changes in the nuclear shape. Is that what you saw as part of as a response to stretch in the same way that you do see with stiffening as well?
So we see changes in nuclear shape and size in both the wild types and the mutants. But I'm sorry, what do you mean about the stiffening?
In stiffening, they have seen changes
Oh.
in nuclear shape, and I was just checking whether you saw the same sort of thing.
I've not had a look,
Okay.
but I'd like to.
Can I ask a question of Warburton? You had the significant differences were between the early infection phase versus the intermediate and late. And you kind of mentioned it in your last slide. If you were able to identify patients who had an early stage infection, does that phase of the OV life cycle open up more therapeutic gaps? Can you target the infection in a different way when it's at that early stage?
Especially early stage.
Sorry?
Could you repeat the question?
So you had very different transcriptome of early versus intermediate and late in the life cycle. Does that difference, if you could identify patients who had early stage infection, is that differently therapeutically targetable? Or not yet?
Not yet to focus on different therapy for different stage of infection. But right now I focus on the the overview of the transcriptome and the gene that could use as a biomarker for the one that can pick up for the biomarker for whole stage of infection. But actually I think that the specific biomarker could be concerned because we obtain quite different genes from different stages. But I have to look further for that again. Yep.
Question on the floor.
Thank you for all your talk. I'm Nitiwat from Imperial College. This question might be toward to Ms. Warapon. A bit mixed with question and comments. So you only taste with the parasite itself, like the fluke, not the adjacent tissue or the host tissue. Okay. If is the case, or if you do you have a plan to taste it at all, or I mean in the host tissue, like a bile duct of the host, mice, hamster. And if yes, sorry, if the answer is yes, what do you think you can see the difference or the similarity in terms of gene mutation or the gene upregulate in the host and in between the host and parasite?
May I sum up your question? You asked me that how can I apply with the human at the specimen, right? Not only in terms of parasite.
Yes. So first question, do you have a plan to also run the transcriptomic analysis in an adjacent tissue of the host where OV habitat to see the difference or similarity in terms of gene mutation, whatever you test or you can find?
I think that is a good plan to do further. But right now we try to understand in terms of parasite first.
Yeah.
But I think if I have a chance to do that, I think in the term of host, it will be give the better understand about the relationship or association between OV or CCA. Yeah, I think that is a great idea.
Yeah, another question is, if you, in your opinion, if you try to test the whole tissue, the adjacent host tissue, what do you think you can detect from that when compared to a parasite tissue that you extract mRNA from that one? Like the gene expression, the immune response, gene about immune evasion, immune stimulate, or do you have any idea, or do you think?
I think.
I know it's in the far future, but I'm just curious.
Right now, no exact plan.
Yeah. That's what bringing to what my comments as well. It could be interesting to see if we can run it in parallel and see the different or the similarity in terms of gene response or gene mutation between the two tissues. So we could understand better in terms of the way the fluke or the host interact with each other, and then we can find a prevention or treatment in the future. And then that's my comment.
Yeah, thank you.
Okay.
If you've got a question.
So, Sheila,
Or
final question, a short answer, please, Sheila. How far away are you from the human experiment? And does the experience of breast cancer help you? Because that's the only place where any of these CDK inhibitors are being used.
I think we need to have a conversation, if we can, John, about the human experiment and where we can go. It is being used, niclosamide is being used for prostate cancer in phase 2 trials at present. Hasn't been very efficacious because I believe absorption is a problem. And so there does need to be some modification of that compound so that it gets better absorbed, essentially. So, yes,
A little way off.
a little bit.
Okay.
Cool, thank you. So we've got just over 10 minutes for tea now, and then we'll reconvene and hand back to Rachel and Heinz Josef to take us to the close. So we'll finish by thanking our speakers for this session.